Um, is this being recorded? Is this being recorded? Um, let's get started. It's my great pleasure to introduce Marlene Cohen for today's seminar. And Marlene has been always at the forefront of correlating neuronal population activity in the visual system with perception and behavior. She graduated from MIT majoring mathematics and brain science, brain and cognitive sciences. And then she went to Bill Newsome's lab to start exploring this question. And since then, she has been working on this. And she worked on the question of how single neuron activities relate to psychophysical performance of monkeys, studying activity of neurons in middle temporal area, which is the center of motion related activity in the monkey brain, primate brains. And there, so this is kind of the most developed system in which where people can say very rigorously about how single neurons activity give rise to a perception and behavior. And there was some big mystery in the field at that moment, which was single neurons are often as sensitive as monkeys' performance, or even more sensitive than monkeys' performance. The puzzle was why single neurons are so sensitive. Why can't the brain average the activity and get better performance? And Marlene's work in the graduate school actually looked at this paradigm, this experiment in reaction time version of the task, so that constraining the time window during which the activity could be pulled. And she elegantly showed that if once you take that into account, then the neuron sensitivity decreased, so that partly explains why neurons appear to be more sensitive. It was just longer than the monkey actually used. And then, and after that, she moved to another movement-related physiology expert, John Manzel's lab, at the time when he was in Harvard Medical School. And there Marlene studied how attentions modulate activity of neurons, in particular paying attention to noise correlations. I think noise correlations come from the studies when Marlene was studying in the Newsome's lab, but she looked at specifically how attentions modulate that, and she did very elegant work. And she continues to work on the similar project, and she is really developing ideas, most recently collaborating with Byron Doiron, and incorporating a lot of theories. So I think I'm looking forward to hearing her recent work. Thank you for coming.
So thank you so much now for the kind introduction and for inviting me here. As now mentioned, life has taken me to Boston, both as an undergraduate and then as a postdoc, and it's fun to be back. So last night, on the way back from the airport, I stopped at Mary Chung's in Central Square, which I loved at first as an undergrad and then later. And I walked in to get my takeout, and the waitress looked at me and went, "Where have you been?" So here I am. I hope you'll indulge me for 1 minute. I will show you a picture of one of the other projects I have been working on since I left Boston. This is a picture of my younger son, Aaron, who just turned 3. He came to work with me the other day and found this big stick that we use as a pointer. I kept walking around to the posters on the wall and going, ta-da! So I think that's going to be my new presentation style. But he goes to a daycare at CMU, And they every day send us an email that sort of chronicles what they've talked about in their meeting time, which is these people are saints or something. But recently I got this email that they had talked about biology, and they had gone around and asked the kids the kinds of advanced biology questions you ask 2- and 3-year-olds. You know, like, what part of your body do you use to hear? And this one little girl said your ears. And what part of your body do you use to walk? You know, your legs. And then they asked Aaron, what part of your body do you use to see? And he said, my visual T-Rex. And so we have a little bit of clarification to do on T-Rex versus cortex. But he is right, of course. And that is because we have long known, and a lot of visual and sensory and cognitive neuroscience has focused on the fact that what you see is what you perceive, what you're able to act on, is often very different than what comes in through your eyes, than what's encoded in your retina. So if I show you this picture of the University of Pittsburgh on a nice fall day, you could look at it for a long time, much longer than you typically look at things. So we as primates move our eyes about 4 times per second, but now you've been looking at this picture for much longer than 250 milliseconds. But still, if I take it away, you could probably tell me some things about it. You could tell me what you thought was important. Maybe there's this big cathedral-looking building in the middle of it, that there were fall colors and some trees. But very few of you could tell me how many trees there were, or whether there were people in the scene, even though that information is encoded in your retina beyond a shadow of a doubt. And that is because we all focus and select the most important aspects of a sensory scene for further processing. And that process of selecting the most important, in our case, visual information for perception and action, we call attention. And my lab studies attention and other cognitive processes and how they affect perception, because we really think the cognitive part is very fundamental to how we see in the first place. And we also think it gives us a handle on basic principles of neural coding. So our idea is that if we measure what changes in the brain when something is important to you versus when it's not, or when you can see and remember and act on that information versus when you can't, we think that will tell us something fundamental about what's important about the neural code in the first place. So in the lab, we study a particular form of attention, which is because it's easy to study in the lab, and that is spatial attention. So all of you, just like the rhesus monkeys who are the subjects in our experiments, have the ability to look at one location, but pay attention to another location. For these guys, making direct eye contact is a very aggressive act. It's a show of dominance. And so a younger, more submissive monkey will almost never look directly into the eyes of a bigger, more dominant animal. But we can all look at this picture and know that even though this guy is staring straight ahead, we have a really good idea about where he has allocated his spatial attention. And the reason he does that is presumably because he is familiar with the more than 400 years of psychophysical research showing that when you pay attention to a location, you're better at seeing things in that location. You are both faster and more accurate at detecting changes in a visual stimulus, and that's very adaptive in certain situations. So in the lab, instead of bringing in bigger monkeys to scare our little ones, we train them to do tasks for a juice reward, where they learn that if they allocate their attention in different locations, they'll get more juice. And so here is an example of one of these tasks, and one that I'm going to talk about today. So the monkeys sit in front of a computer monitor. These rectangles show you what's on the monitor at different points in time. A trial begins when the monkey fixates a central point of light, and he's required to do that throughout. And then 2 moving Gabor stimuli, these stripy things, start flashing on and off. And at a random and unsignaled time in the trial, the motion direction, and therefore also the orientation, of one of these stimuli changes slightly. And the monkey's job is to tell us that he noticed that change by looking at the stimulus that changed within a few hundred milliseconds. We can manipulate spatial attention in this task by cueing the monkey in blocks of trials as to which of those stimuli is most likely to change. So if it's an attend left block of trials like I've drawn here, the monkey has learned that the stimulus on the left is going to be the one that changes on 80% of those trials. And he's rewarded for making an eye movement to either stimulus that changed, whether it was on the cued or the uncued side. And when we do these sort of manipulations, we see that attention has a huge effect on his performance, and so therefore the amount of juice he earns. So this is an example psychometric curve from one experiment. This is the monkey's proportion correct, detecting motion direction changes of different amounts. You can see that when the change is very big, it's very easy for the monkey. He essentially always gets it correct. When the change is very subtle, he almost never detects it. And middle performance for intermediate changes. This is the 20% of trials where we tricked the monkey. We told him the change was going to happen on the left, and it happened on the right instead. And you can see that for this example experiment, attention, knowing where to expect the change, improved the monkey's performance from something like 40% correct to nearly perfect performance, right? So that's a really big deal if you are a monkey interested in earning a lot of fruit juice, okay? And so our goal here is to understand what is it that changes in the brain that causes these big improvements in performance. And so broadly, I'm going to tell you about some work from my lab asking broadly how cognition affects perception. And by perception, I mean performance in the visually guided tasks like the one I showed you. I'm going to start with this question of which spatial attention-related changes in the neuronal responses affect behavior. The answer to this is that it's going to be a mechanism that we think is new, that we haven't seen in the literature before, that involves the activity of whole populations of neurons. So then I'm going to ask what the relationship is between those population mechanisms and the more well-studied, well-known effects of attention on one or two neurons. And then finally, I'll talk about some new and ongoing work in the lab, asking how general these results are and whether the population mechanisms associated with spatial attention could explain other cognitive processes. Okay, but we're going to start with spatial attention. So my lab is far from the only one to study the neural basis of spatial attention. In fact, if you type neural basis of spatial attention into PubMed, you get something like 10,000 hits. And so I'm going to summarize those. So this is a very nice schematic from a review by Bob Wirtz showing many of the brain areas that we think are important for tasks like ours. Those include a variety of brain areas in visual cortex, including the middle temporal area that Now mentioned, which we think is important for motion processing. And then also a network of interconnected oculomotor areas in the parietal lobe, in the frontal lobe, and in the brainstem, which we think are important for perceptual decision making and have also been hypothesized to be sources of attention. And the summary of those 10,000 papers is that when you teach monkeys or humans to do an attention task, and you stick an electrode or you measure an fMRI bold signal or some other measure of neural activity in any of these areas, you see an attention-related change in whatever your favorite measure of neural activity is. Those range from changes in mean firing rate of individual neurons, changes in bold activity, changes in timing of spikes, in synchrony, in oscillations in local field potentials. And so a huge variety of effects of spatial attention have been characterized. But it's been difficult so far to really connect those changes, which have been replicated many times, to those changes in behavior. And I am hopefully going to convince you that we can make new traction by recording from many neurons at once to give us the power to understand behavior on individual trials, from recording from considering multiple brain areas at a time, and also by constraining our analyses by the animal's behavior. And so the project I'm going to talk about first was led by a terrific postdoc in my lab who is a graduate of the Harvard program in neuroscience who did his thesis work with Rick Bourne. That's Doug Ruff. And Doug recorded from groups of neurons while monkeys performed the spatial attention task that I told you about before. He recorded from groups of neurons in two brain areas at the same time. MT, which is this extrastriate visual area that we think is responsible for the perception of visual motion. So we think of that as sort of the sensory input to our task. and also the superior colliculus, which is a brainstem nucleus that's been long known to be involved in both vision, planning eye movements, and then more cognitive phenomena like decision making and as a source of attention. So if you microstimulate, if you put in a little bit of electrical current in the colliculus, if you put in a lot of current, you'll move the eyes. If you put in a little bit of current, you mimic both the behavioral effects of spatial attention and the effects on visual cortex. So what made this experiment difficult is that Doug went in every day with a multi-electrode probe, and one of those probes in each of these two areas, and hunted around until he found groups of neurons with largely overlapping receptive fields. So these are the centers of the receptive fields for one example experiment. This is a map of visual space. The monkey's fixating here. The red dots are the centers of the receptive fields of the MT neurons he recorded that day, and the blue dots are the centers of the receptive fields of the colliculus neurons, and these circles are the median size and location of the receptive field in each area. So we have groups of neurons with more or less the same spatial receptive field, and Doug has monkeys perform that task where one of the two visual stimuli is in the receptive field of these neurons and one is far away. And so we are measuring what happens in the brain when monkeys switch attention into and out of the receptive fields of these neurons. So the question is, neuronally speaking, how does spatial attention improve perception? What accounts for that really big improvement in performance that I showed you? And if you think about this from the monkey's point of view, his job is to use the activity of neurons in his brain to differentiate between these repeated presentations of this original stimulus. I'm going to color code this in orange in the next few slides. And then to tell when the thing has changed. So that's going to be my blue stimulus in the next few slides. So he needs to use the activity of his neurons, say in visual cortex, in MT, to tell the difference between those two stimulus, those two directions of motion. So if we think about what we know about the activity of MT neurons, we know that they respond differently to stimuli that are moving in different directions. And so we could schematize the response of a population of neurons to these two stimuli, the orange one and the blue one, in this way. So I've labeled the axes of this cartoon as MT dimension 1 and MT dimension 2. You could think of this as the activity of two simultaneously recorded neurons in MT, neuron 1 and neuron 2. In a minute, I'm going to ask you to think of these as principal components in the high dimensional space where each dimension represents the activity of one of the neurons that we recorded at the same time. Okay, so these are just sort of activity axes. We know that MT neurons respond differently to different stimuli, and so we might plot the response to that orange stimulus here. We know that the responses of neurons are variable, so when you show that stimulus a bunch of different times, you get a slightly different response on every trial. Schematize that with this oval, and then we might get a different response to the second stimulus. And then when we think of the process of decision making as converting this population activity into a decision about how to act. And because the decision here is binary, either the monkey makes an eye movement or he doesn't, then we could think of this as sort of a one-dimensional process. So maybe the responses of the MT population are projected onto one dimension. I've called this the decision axis. And that dimension may or may not be optimal for dividing distinguishing between these two responses to the two different stimuli. Okay, so maybe this is our cartoon for what happens in the unattended case. And we know, I already showed you, that the monkey does much better at our task when he directs attention to the receptive fields of these neurons. And so the question is, what is changing about this story that might cause this improvement in perception? The first hypothesis, which I think in one way or another has motivated most work in the field, and especially my work in this field, is that attention improves information coding. We know that when you pay attention to something, to a stimulus, you see it better. And so maybe that's because in your MT, in your visual cortex, the attended stimuli are represented better. And what that might mean here is that the representations of the orange stimulus and the blue one become more different. Okay? Another hypothesis that has motivated work in the field, and these two hypotheses are not mutually exclusive, is that instead of or in addition to improving the way those stimuli are represented, you could improve the way that they're read out to guide behavior. So maybe you change this decision axis so that you more efficiently read out the information that's there, and that could lead to an improvement in performance. And so what Doug did was use his,
So this seems to be the worst decision.
Yes, this is the axis that you might project these responses onto to better. So the hyperplane dividing these two would be orthogonal, yeah. Yes. Okay, so the first thing that Doug did was to just look in his data to see if he had evidence for either of these or both of these mechanisms. So let's look at hypothesis 1. Okay? So the thing he did to test this was simply look at his population of MT responses and use a linear decoding strategy. We also experimented with nonlinear ones to ask, does attention make the representations of these two stimuli more distinguishable? So here is a schematic of this process, which I'm going to walk through because I'm going to modify it slightly in a minute. This is a matrix of the population of MT responses, in this case, projected onto the first 10 principal components. We did this just to limit the number of weights we needed to find. That doesn't matter for anything I'm going to talk about today. So you could think of this as the responses of 10 neurons, in this case 10 principal components, 10 axes, on a bunch of different trials, different stimulus presentations. The gray scale here is the magnitude of the response. And these are sorted by the motion direction, whether it was that original, that first stimulus, or the one that changed. Okay? And what he did was use simple linear methods to find a set of weights that would best allow him to differentiate between those two directions of motion. To make it cross-validated, he then took a separate set of trials, took a linear combination using these weights, and then asked, how well can you distinguish motion direction from that population of MT responses? And did that separately for the two attention conditions. So now we can ask, does attention improve information coding? By asking, how well does our linear decoder do distinguishing those two stimuli when the direction change was very subtle? So this is an intermediate direction change that the monkey gets right sometimes and gets wrong other times. And so we can plot performance here. I called this the stimulus decoder. And ta-da, it's a not very impressive result, right? So what we see is that there's lots of stimulus information in MT. That's good. But that there is not an impressive, and in this case, not a significant change in the ability of this linear decoder to distinguish those two stimuli in the two attention conditions. Yeah.
How do you measure the unattended case in the experiment? This is from the 20% cases?
That's right. So it's always comparing, say, stimulus changes that happen on the left inside the receptive field of the neurons, but it's when those changes were in blocks where he was cued to attend to the left versus blocks he was cued to attend to the right. So,
but is the Q 100% valid?
the Q is 80% valid. So it's the 20% of trials. Exactly. Yes.
But it's like disattended.
Yes, sure. So we just have these two conditions, and we can't and don't make any statement about where it is versus neutral. So that's right. Yes. Better to say cued versus uncued in this case. Yes. Okay. So as I said before, this was the hypothesis that really drove a lot of my work. I thought maybe, I don't know, Doug messed up the neurons somehow. We went back to a lot of old data sets, including data sets from other visual brain areas using the same task, and also from different tasks, in particular a discrimination task, because we wondered whether there was something special or weird about change detection. And the story was really the same. In some of the, I think we analyzed data from 18 monkeys altogether in 2 different tasks and 3 different brain areas. And the story was always that there was a very small, sometimes significant, sometimes not, effective attention on the ability of these linear decoders to distinguish motion direction. It was in no cases correlated with the amount of behavioral change from day to day, and it was in no cases big enough to explain the monkey's change in behavior. Yes?
Was there any improvement in information coding due to learning?
That's a good question. So we have data about this. These data are all in very well-trained monkeys. We have data about learning. It's a little bit tricky to make those statements because we're not sure whether we're recording from the same neurons throughout the course of learning. To a first approximation, there was not an improvement in information coding due to learning. There was not. Yeah. Yes.
Marlene, sorry.
Yeah.
Do you have a handle just behaviorally whether the animals are, whether attention is affecting in this task D prime versus criterion? Sort of
Yeah. It is, so we do not explicitly control that the way that John has nicely done. In all of these data sets, attention is affecting both. And in these change detection data sets, they really go up and down together. So I think in this data set, the attention-related change in sensitivity and criterion were correlated on a session-by-session basis of like 0.85. So these data just don't allow us to, so you get an increase in sensitivity, you also get whatever you call it, a more liberal criterion, and we can't parse those out in these data sets. So John's work makes you think that this is mostly related to sensitivity, but it's hard to know. Yeah. Okay, so bummer. So then we started to look, well, maybe these other people are right. Maybe what attention is doing is changing the readout such that the visual information in MT to a first approximation isn't changing, but somehow it's being used more efficiently to guide behavior. Okay? So to test that hypothesis, Doug used a really similar decoding strategy. So this is what I showed you before. We called this the stimulus decoder. I will put it up there. Now we wanted to know how the visual information is actually used to guide behavior. And so now, instead of finding the set of weights that best allowed us to distinguish motion direction, instead, we found the set of weights. We tried to ask, what's the monkey's decoder? And so we found the set of weights that best allowed us to predict the monkey's choices, the monkey's behavior. Because the monkeys were not perfect, that was related to, but not exactly the same as motion direction. So on average, when the stimulus didn't change, he didn't make an eye movement, and when it did, he did, but that was not exactly the same. So we found a set of weights that was optimized to let us predict the monkey's choice. But then our question is still about motion information. So we asked, okay, we found our weights that we used to predict the choices, but now in our same cross-validated set of trials, we asked, using these weights, how much motion information was there? So the question is, how much stimulus information, just like up here, is there in this case that could be gleaned from an optimal decoder, in this case that the monkey seemed to be using to guide his behavior. And then a third kind of decoder that we used is that we made use of the fact that Doug recorded simultaneously from two areas. And so we set up a set of weights that were designed to capture the information that is shared between these two areas. So we called this the colliculus decoder. And now we find a set of weights that best allows you to predict the matrix of colliculus responses from the matrix of MT responses. Now there's a linear classification step, but we use the same idea to ask how much visual information is being shared between those two areas. Okay? And again, we do this separately for the two attention conditions. And so by definition, this is the optimal decoder. These ones do worse by definition. But the question is, are there effects of attention on the amount of visual information that's either used to guide the choices or that's shared with the colliculus? And the answer is yes. So here is what I showed you before. Here is the performance of the amount of visual information that was gleaned by this choice decoder or by the colliculus decoder. And what you see is that in both of these cases, first of all, they do worse than the optimal decoder. That had to happen. But you see that there are bigger effects of attention, and more importantly, the effects of attention on the choice decoder and the colliculus decoder are bigger than the optimal decoder, and they correlate on a day-by-day basis with the monkey's behavior. I'm not going to walk through that because I want to tell you about something I think is even more important, but the idea is we kind of buy this a little bit more because the attention-related change on one recording session that we come up with when we use these methods is correlated with the amount of behavioral improvement that we see. So when the monkey shows a bigger behavioral effect of attention, you see bigger effects of attention on the amount of stimulus information used to guide choices or shared with the colliculus, but not on the amount of stimulus information that our optimal decoder could bring. Yeah. We do. It is pretty small and difficult to analyze, and that's not because, so that's related to Rick's question about criterion. It is difficult to analyze neuronally because there are not very many of those trials. So the structure of our task, I think we train the monkeys to be very conservative because on a lot more trials they need to hold their eyes still than they need to make an eye movement. Attention does make them less conservative, but we only have a handful of trials to analyze. Okay, so what Doug realized is that these results are not especially consistent with hypothesis 1, that attention improves information coding. They are consistent with hypothesis 2, that attention changes the decision axis. But we realized that they were also consistent with a hypothesis that we haven't seen in the literature before, which is that we know from our work and the work of many others that attention has pretty big and replicable effects on the responses of MT neurons. That was true in this data set as well. And one possibility is that attention changes what's going on in MT, not so that the two distributions are more separable by an ideal decoder, but instead so that they line up better with this relatively fixed decision axis. Okay? So the difference between these two hypotheses is that in this case, you've not changed MT, but you've changed the decision axis. In this case, you've changed what's going on in MT, but you have not changed the decision axis. And of course, these are not mutually exclusive either. And so to differentiate between these, to see which of these seem to dominate our results, and by dominate our results, I mean account for that attention-related change in the choice decoder and the colliculus decoder, we did this weight swapping procedure. So here is our rationale. What we did was we went through and found the weights for the choice decoder and the colliculus decoder exactly as we did before in the two attention conditions. But then we traded. So our reasoning was, if what is happening, if the attention-related improvement is due to what's going on in MT, then if we use the MT responses from the attended condition, it doesn't really matter which weights we use because maybe they're not changing, and we should see better performance, more stimulus information. On the other hand, if what's happening is a change in the weights, an improvement in the weights, and an improvement in the decision axis, then what should matter is that we need the weights from the attended condition, and maybe it doesn't matter whether the MT responses come from the attended or the unattended condition. And so if I plot now the performance of our decoder, the amount of stimulus information that our decoders can decode, using the MT responses from the attended condition with the weights, the decision axis from the unattended condition, versus the same performance, but now using the responses in MT from the unattended condition and the weights from the attended condition. If what matters is what's going on with the weights, the change in the decision axis, we should do better when we have weights from the attended condition. Our points will lie below the diagonal. On the other hand, if what's happening is a change in MT, not a change in the weights, but now a change in the MT responses, we need those responses from the attended condition in MT, and our points should lie above the diagonal. And to a first approximation, that's what we saw. So these are the results using the choice decoder and the colliculus decoder Each point here is the result from one experimental session. And what you can see is that most of them lie above the diagonal. And what that tells us is that to a first approximation, what's happening is a change in MT. I showed you that that's not well explained by an improvement in information coding, but instead it's a change in MT responses so that they seem to better align with a relatively fixed decision mechanism. Okay, so this was a surprising result to us, not just because it sort of went against our hypotheses, but also because this seems very exotic, right? I'm saying that there are these changes in MT, they're not improving information coding, there's some weird like reshaping, reorienting, rotating thing going on so that they're better aligned with a decision mechanism. And what we wanted to know is how that relates to those 10,000 other studies, and in particular, how that relates to the well-studied effects of attention on just a couple of neurons, which was the history of this field. So when I drew this new hypothesis, I drew a couple of different changes, right? I said that in the schematic, I made attention change the mean responses of these neurons so that it changed the mean position of those clouds of points, those ellipses. And I also kind of squished them in different ways, right? And we know that attention does both of those things. And as now mentioned, a lot of my previous work, including my work as a postdoc with John Mansell, was focused on sort of the shape of these ellipses, what attention does to the correlations in the trial-to-trial variability in the responses of neurons in visual cortex. And so we focused on a particular measure of these shared fluctuations called noise correlations. They're also called in the literature spike count correlations, or RSC. Those words all mean the same thing. And they measure the extent to which, if you record from two neurons at the same time, the trial-to-trial fluctuations in their responses are correlated. So you could imagine that if we record from two, say, MT neurons at the same time, we could show the monkey the same visual stimulus over and over again, count the number of spikes that neuron 1 fired on each trial, neuron 2 fired on each trial, so now each point is the response of these two neurons on one trial. It might be that the trial-to-trial fluctuations in those responses are highly correlated. We would summarize that with just a Pearson correlation coefficient. But you could imagine doing the same experiment and finding that the fluctuations in the responses were completely uncorrelated. What John and I found, and what many studies have replicated since then, is that in visual cortex, we know that the responses tend to be correlated, not nearly this much, but somewhat positive. And what attention does is decrease those correlations, take you from a situation where the responses are rather highly correlated to where they are not. And what Amy Nee in my lab found recently is that the extent to which that happens, the change in the average correlation coefficient in a population of neurons, was really predictive of the monkey's performance. And so what this plot here, I should say Amy is also a wonderful postdoc in the lab, and she also is a graduate of the Harvard program in neuroscience. What she found, what she plotted here, is the average spike count correlation between pairs of simultaneously recorded neurons in a different visual area, in area V4, while monkeys perform the same task. And on the y-axis is a measure of the monkey's performance. This is that sensitivity measure. Big numbers are good performance. She color-coded these points by whether this was when the monkey was doing, the change occurred in the attended versus the unattended condition. But what you can see is that overall, there's this pretty strong relationship that when correlations are low, the monkey does well. When correlations are high, the monkey does not. And so what we wondered in Doug's experiment is whether that improvement in the amount of stimulus information that was used to guide the decisions could be accounted for by these changes in spike count correlations. So the first thing he did was redo that choice decoder, but this time, our idea is that maybe the weights, the decision axis, doesn't change with attention. So he used one decision axis, one set of weights in both attention conditions. And what you see is that when you do that, you still decode motion information just fine, and you still see this attention-related change in the amount of information used to guide the monkey's performance. And so then, to ask whether those changes in correlations could account for that improvement, he decomposed the MT responses into their mean responses to each stimulus and in each attention condition. So these are the means plus the residuals. And the residuals account for that trial-to-trial variability. It accounts for both variability that's private to each neuron, in this case, each principal component, and also shared between them. And then he swapped them. So the idea is, if the attention-related improvement is due to the change in those residuals, the change in correlated variability, then you should be able to take the residuals from the attended condition and even combine them with the mean responses in the unattended condition and still do just as well. And that's what he found. So when we use the residuals from the attended condition, but the mean responses from the unattended condition, you get a performance that is indistinguishable from the full responses in the attended condition. So this tells us that what's happening is not improving information encoding, not changing the decision axis to a first approximation, but instead this change in correlated variability accounts for this realignment, so that the amount of visual information that is communicated, that is either used to guide behavior or is shared with the colliculus, is improved with attention. Okay? So if we put this all together, I think we have a story emerging here. Our group and others have found that these changes in correlations are very specific. It's not just that you reduce correlations among each pair of neurons in different ways. Instead, attention affects correlated variability in just one dimension in population space. Many groups have found this. This is our demonstration in a paper with Brent Dorrell. So we used, in this case, factor analysis. You could think of this as principal component analysis to find the dominant dimensions that account for the greatest shared variability between the neurons we recorded from. And what is plotted here is the amount of shared variance, the eigenvalue for those of you who do this. And what you can see is that attention changes the amount of shared variance, but it really only does that in that first dimension. Put another way, if we look at the raw covariance, these pairwise correlations between pairs of neurons, covariance here not normalized, what you can see is that there's this big attention-related change I've been talking about. But if you subtract out this first dimension and look at the residual covariance, essentially all the covariance goes away and it's not modulated by attention. So okay, so this says attention has effects on mostly on one dimension. And what Amy found is that that very specific dimension seems to be the choice decoder, seems to be the one that the monkey is using to guide behavior. And so what she's plotting here, in this case from her V4 data set, but we replicated this in Doug's MT data set, is the performance of a decoder. This is our stimulus decoder that I've been showing you as a function, how much stimulus information is there, in this case in V4, as a function of the number of dimensions you give it to work with. And what you can see is that this grows with the number of dimensions. Here's the maximum if you use the full data set of neurons she recorded. But if you do the very same thing and ask how well can you decode the monkey's choices, what you see is that all of the choice predictive information that's extractable from visual cortex using linear methods is present in that first dimension. Okay? So I think overall we have this story. Attention, and then it turns out other cognitive processes too, affect correlated variability along one dimension. This seems to be the dimension that the monkey is using to guide behavior, and that accounts for the attention-related improvement in the stimulus information that's used to guide choices. Okay, and so in the last few minutes, I'd like to tell you about some very new and ongoing work in the lab. there are some reasons to think that spatial attention might be special in certain ways. The biggest one is, as I told you, all these experiments are designed with neurons with overlapping receptive fields. The monkey is directing attention into and out of those receptive fields. And so what's happening is that attention is making those neurons relevant or irrelevant. Right? But a lot of what we do in vision involves the same populations of neurons, but extracting different information from them. If I show you this laser pointer, I could ask you what color it is, and I could ask you what orientation I'm holding it in. And you would use, to a first approximation, the same populations of neurons to answer those questions. And what we wondered was whether task switching in that way, decoding different visual information from the same population, could be possibly accounted for by this same funny reshaping mechanism. This seemed like a situation to us, if you're ever going to change that decision axis, then reading out different information from the same population would be the situation where we'd see it. Okay, so our question is, how does the brain extract different visual information from the same neural population? This work is led by another terrific postdoc who has no connection with Harvard that I know of, named Cheng Shi, in the lab, with terrific help from a post-baccalaureate student named Lily Kramer. And what they did was teach monkeys to do two different visual discrimination tasks. So in this case, they showed two Gabor stimuli, one after another. The second one was different from the first in two ways. It was shifted either to the left or to the right. And its spatial frequency, sort of how wide or narrow those stripes were, increased or decreased. And in alternating blocks of trials, the monkey was asked to make an eye movement to report whether the thing moved left or right, if it's a position trial, or to report whether the spatial frequency decreased or increased. Okay? And so our question is, so we have now the same eye movement, which is associated with two different stimulus features in the different versions of the task. And so our question is, does the decision axis, does the way that visual information is read out from visual cortex depend on what feature the monkey is reading out? And in this experiment, they recorded from two different visual areas that we think are important for these features, primary visual cortex, or V1, and area V4. So our question is, does the readout, does the decision axis depend on the task? And the way we're going to ask that question is to ask, how well can we predict the animal's choices on one task using the weights from that same task or from the opposite task? So if I want to predict if it's a position discrimination task, and I want to predict whether the monkey is going to tell me that the thing moved to the left or the right, I can use weights to best allow me to predict his choices on that position task. Or I could instead find the weights that allow me to predict whether he's going to tell me whether the spatial frequency decreased or increased, and apply that to trials from the position task. That makes sense? So I'm going to show you fake data first. It's in red so that I remember to tell you that it's fake. Okay, so you could imagine that the weights, the way that the monkey reads out information from these visual populations, depend very much on the task. So maybe we can predict the monkey's choices pretty well, whether he's going to say the thing moved to the left or the right, using the weights from that same task. So maybe these points are all above chance or mostly above chance, which would be 0.5. But then the monkey uses totally different weights, a totally different decision axis, to discriminate spatial frequency. And so now our ability to predict whether he said the location moved from left to right is essentially at chance when I use the weights used to predict the spatial frequency task. Does that make sense? Another possibility that would be more in line with the spatial attention results is that the monkey uses the same decision axis in the two tasks, but maybe something is changing about visual cortex. And so now the weights from one task should allow you to, from either task should allow you to predict the choices about equally well. Okay. Yeah?
So is that assumption hold when basically the decisions are independent? Is that fair to assume here?
Yes, and so that's a really good question, and I can tell that from the animal's behavior. So I'm not sure I have this slide, but so if we ask, say it's a location task, I can ask how does the probability of him looking at the target on the left or the right depend on the spatial frequency change? And it took a lot of training, and to be perfectly honest, I thought it was going to be impossible, but I have monkey whisperers in the lab, and so those lines are flat. But yeah, this is a really important point. Okay. So to a first approximation, I should say, actually, this is data just from one monkey, so this is very early days. But our results look an awful lot like the attention results. It looks a lot like the monkey is using the same weights in both tasks. So here is now real data from area V1. You can see that we can predict choices above chance most of the time. And this is all cross-validated, which is why sometimes you can veer below chance. But you can see that to a first approximation, you can predict choices, say, on that position task equally well using weights from the position task or from the spatial frequency task. And the same is true in V4. Okay? So maybe the takeaway from all of this together is that a fairly simple mechanism, changing neural responses maybe by changing correlated variability along one axis, causes changes in the visual information that is used to guide behavior and is shared with downstream areas. And that really simple mechanism maybe can give rise to pretty complex cognitive phenomena, including attention, including task switching. And we've wondered in the lab whether simple mechanisms in general can explain even more complex behavioral phenomena. And so the last thing I will show you is that our lab has had a long-standing interest not just in attention, but in disorders of attention. And as a step towards studying those, we've started to ask whether very simple manipulations, such as those used to treat disorders of attention, can give rise to specific cognitive phenomena. And so Amy, as well as another post-baccalaureate student named Brittany Bowes, have started giving stimulants to our monkeys. They have focused mostly on two drugs that are used to treat disorders of attention. Methylphenidate is the key ingredient in Ritalin. Amphetamines are the key ingredient in Adderall. And then this is my favorite performance-enhancing drug. And what they did is they gave these things to the monkey. The most important result of this experiment is not what I'm going to show you data for, but the monkeys, when they are given Ritalin and Adderall, work 3 times as long. So this is everybody's favorite experiment in the lab. The caffeine, incidentally, has a very little effect on that. So I don't know what that says for those of us who tried. But the important thing I want to tell you about is that this very non-specific manipulation, so these guys just mix these drugs into juice, so this is a systemic administration. There's no like micro-infusing into specific brain areas or anything. But it has a selective effect on spatial attention in the task I've been telling you about. What they find is that if you plot the monkey's average performance when they are on these drugs versus their average performance when they are not, these drugs tend to improve performance at the attended location. So in this case, each point is the average for one monkey and one drug. And they tend to lie above the diagonal, which means that performance at the attended location is better when they are when they have had these stimulants. But that is not a general improvement, like the monkeys just are better across the board. These drugs do not improve performance at the unattended location, and in fact, some of them make performance worse at the unattended location. I can show you that in a slightly different way. Now each point here is an experimental session for the 3 drugs, and what you can see is that the effect of attention, the amount that attention affects performance, is bigger when they are on these stimulants than when they're not. So these points tend to lie above the diagonal. So we have a situation where we have a very non-specific manipulation, giving drugs by mouth, that has a selective effect. And so we're kind of interested in and some of our modeling efforts are going toward trying to understand how very simple general mechanisms, maybe like the ones I've talked to you about today, can give rise to sort of specific cognitive phenomena. Okay, so I'm just going to wrap up. I think that going forward, I and a lot of people feel that the keys to making progress linking neurons to behavior are going to be really measuring behavior very carefully and recording from many neurons in many areas at the same time. And I think the key for us was that we really didn't get that far when we just analyzed the neural responses in isolation of the behavior, that really constraining our analyses by the monkey's behavior or by the activity of downstream neurons was key to understanding what was going on. I showed you some evidence for this hypothesis that attention improves performance mostly by aligning neural responses to a relatively fixed decision mechanism. And we have some early evidence that that might be a general feature, and in particular that simple mechanisms might account for complex cognitive flexibility. So I would like to end by thanking the members of my lab and thanking all of you for your attention.
Two questions. One, so a simple interpretation of the results would be that basically the monkey is using for the decision basically differences in the total spike count, and this is where most of the correlated variability is. So it's a very suboptimal axis that more or less would explain the results. So then this might suggest a manipulation of the experiment in which essentially you can balance the amount of activity. So this will be a very bad, very poor decision.
Yeah, so I thought a lot about that possibility. It turns out that if you actually use the average activity, it predicts the monkey's choices very, very poorly. And what instead I think is going on is that this first axis of the correlated variability, we know that, for example, in MT, if you have two neurons, you can predict the extent to which their responses will be correlated by their similarity in a lot of different factors: how similar their receptive fields are, how similar their preferred directions are, their tuning for color, for speed, motion speed, all these things. And in our simulations, at least, the axis that explains the most shared variability, if it depends on many, many features, is actually the axis that is best for discriminating natural scenes. So if the correlated variability depends very much on signal correlation, but over a very broad stimulus set, then that axis is actually very close to the optimal axis. It is not optimal if the monkey knows that he's only ever going to be seeing black and white Gabors all day long, which he should because he has done this task an awful lot of times. But if you are just discriminating motion direction and you were doing that for any stimulus, including a moving banana or whatever it is, then that axis is fairly close to optimal, and it turns out to be different enough from the just average activity axis.
So one second question is about your point about multi-area. I didn't seem to be convinced that you actually gain something from multiple areas. It seems to me that you basically got the same conclusion from here.
That's right. So I think there are two things we gain, and I didn't talk about them very much. One is that in our task, the behavior is binary, and so your ability to assign weights to best predict the behavior, the error function is very shallow. So the many possible weights allow you to predict the behavior just about as well because the behavior is not a strong constraint. And so what we found is that the colliculus decoder, we kind of had more to say about the properties of that decoder. And then the other thing I think is going to be important is that there's a lot of really exciting work going on here and elsewhere looking at some very analogous questions in rodents and other animals that you really might not want to trained to the extent that you really believe that when they make an error, it's because of their visual cortex. And there, I think that showing that you can draw a lot of the same conclusions by looking at stimulus information that's shared between areas, even when you don't want to analyze the behavior on a trial-by-trial basis, I think is going to be important for generalizing these results. Yeah.
I'm still trying to get my head around the last result before the drug study. And it may have been, I just wasn't maybe caught up to the question that was asked about independence, but it seemed to me that I wondered about the choice of the two, position and spatial frequency, because certainly to a neuron with a small receptive field like in V1, the two tasks could cause similar changes in the population dynamic. So I guess I'm wondering if you had chosen two tasks that were more different, do you think you might not have seen the same high degree of cross-coder reliability?
I don't know the answer to that because we didn't do it. But I will say that position and spatial frequency are very separable in these populations of neurons. So ideal decoders of each feature are not related to each other. So you don't sort of automatically decode one when you decode the other. I think the bigger criticism is that I think that in V1 and V4 and MT, we only predict behavior so well, and it's not that well. It's well above chance, but it's not that well. And the behavior is binary, so we can't assign the weights very carefully. So I think the bigger concern is you have a mediocre choice decoder. You could have picked any one of mediocre choice decoders. You found a decoder that's equally mediocre for both tasks. And I think that the way to get around that is, I actually think the choice of features is okay, but I think we need a richer behavioral output to be able to nail that very well.
So one question. This attention-based improvement of the task performance should be based on reinforcement learning, right? So at some point, monkeys figure out that paying attention to here makes sense given the task context.
Yeah.
And so also this changing correlate structure of activity may be regulated by different mechanisms, neural observation or top-down input and so on and so forth. Do you have any insights from your experiments what kinds of mechanisms might cause improved implementation?
Yeah. So I would love to know the answer to that question. So that is sort of the motivation for this modeling project with Brent Tarall. And so what they do is create circuit models of attention, and then we use our data to constrain those models. And there are two types of changes that account for the set of neural response changes that we observe. One is that you can change the balance of inhibition and excitation. You could think of that as attention having a bigger input to inhibitory than excitatory cells. Alternately, in addition, you can change synapses, change weights, so that the neurons representing the attended stimulus have a greater impact on the ultimate decision. In these models, those two things work equally well. And we are not seeing, we think, a ton of evidence for weight changes. And I think a really interesting possibility is that weight changes require some longer term process, and that when you're going to switch attention very quickly, you do something like change the activity versus maybe something like learning that's longer term could involve changes in synapses. I would love to know the answer to that.
Okay, thanks so much.
Okay. Thank you.